Hello everyone, welcome to The Living Revolution. We are a synthetic biology podcast introducing you to the very entrepreneurs, thinkers and researchers working in this emerging part scientific discipline, part revolution, holistic approach to using biology. Today we are joined by Victor de Lorenzo to discuss using synthetic biology in fortifying, changing and perhaps saving the environmental microbiome and the steps to scaling bioremediation solutions from lab to field. More on that later. For now, I'll let him introduce himself. Enjoy.
Well, thank you, Sarah, for this nice invitation to contribute to your podcast. Let me start by saying that my name is Victor de Lorenzo. I work in the National Center of Biotechnology in Madrid, that is part of the National Research Council. And we kind of define ourselves as environmental synthetic biologists. And to give you some historical background, we started a long time ago, well, not long time ago, say in the late 80s, early 90s, to think on ways of using genetic engineering for developing agents, for developing bacterial strains in the laboratory, able to deliver some remedies to sites with a history of pollution by urban and industrial waste. So by that time, the tools that were available were kind of primitive. And I have to say that even though the possibility of using genetic engineering to this end was created a big hype, on the short run, it did not succeed too much, right? And but later, because of the onset of synthetic biology years later, and the possibility of modeling and massive DNA, say, synthesis and interactions, all types of tools that have been brought about by synthetic biology. Now there's a new opportunity to really go back to this issue on how to benefit, how to leverage microbiology and the immense potential of the environmental microbiome to deliver activities of interest. How can we nurture that? How can we leverage that for this specific end? So at the moment, my laboratory is really trying to develop the tools to bring this about and to engineer bacteria and agents, as we call it in general, able to be released and then through that release to produce some positive effect in the environment.
Mm-hmm. So you mentioned, you already mentioned just there this term, sort of the environmental microbiome, and you also in your article introduced the term environmental galenics. Could you perhaps explain maybe what both of these terms mean and how they're linked together?
Yes, that's very simple. Well, the term galenics or galenic science comes from pharmacy and pharmaceutical science, and is the whole branch of this pharmacy or pharmaceutical sciences that have to do with delivering, say, remedies to a sick body. Okay, so you may have a specific drug, and you can deliver that through a pill, through a suppository, through an injection, through an aerosol, you name it. So by the same token, so we argue that you can produce in the laboratory a wonderful activity of interest for the environment, say CO2 fixation, degradation of PCBs, this or that. But it's not enough to have the activity. You have to have some kind of channel through which you deliver this activity to the target that you want it to be, effective. And this is why we kind of transplanted this idea of galenics that comes from pharmacy, from the medical sciences, into proposing something similar for the environment. Because sometimes we have this kind of naive idea that it's enough that you produce in the laboratory a wonderful bacterium able to degrade this or to do this or that, but then that's nothing. The big challenge is to scale it up to a dimension that can make a difference, and this is exactly the type of business that we are engaged now.
Mm-hmm. So taking the sort of this bioremediation solutions from laboratory to the actual environments in which they can work through some kind of sort of physical carrier. What are the challenges actually of taking it from sort of this bioreactor scale to the environmental scale, would you say?
Well, it's not only one challenge, it's a kind of list of challenges, okay? And we can go one by one and then try to what degree synthetic biology can help to solve or to address these challenges. The first challenge, in my opinion, is what in synthetic biology jargon is the so-called the chassis. And, in other words, what type of biological container, what type of life agent would be adequate for release. And even once it is released, to go to the right places to produce the activities that are beneficial, making sure that it's safe, that is not detrimental with the native microbiome. I mean, you have all these questions that are connected to the chassis. And then what happens in many cases is that many synthetic biology laboratories put the whole emphasis in pathway development. So you have a number of genes, you put promoters, you put genes, whatever, and then at the end you have a wonderful activity. But that has to go not only to a specific bacterium, but also to something with some physical characteristics. It can be like a rod-shaped bacterium, it can be a small ball, it can be encapsulated, it can be in combination with other things. So this is precisely the type of challenges, in my opinion, or one of the challenges that, in my opinion, we face for upscaling these bioremediation strategies. But there are others. Yeah, go ahead.
Could you tell me more about what these physical containers might actually be like in a physical sense? What are these containers? And also, how does the container change based on the solution that we're trying to solve?
Okay, well, maybe the term container evokes something very physical, and we're talking here about a biological container, okay? At the same time, a biological container is physical as well. So I'll give you one example, so in many cases, biodegradation reactions involve very strong redox processes. And not every bacterium, not every biological system is able to kind of support this type of strong redox transactions. And for instance, E. coli, that that is the most famous bacterium in the world, in many cases is great for the laboratory, but in the environment is completely useless, because the background metabolism in many cases is not sufficient to really support these reactions that are necessary for really making a good job in the environment. So in that case, we have to look for other types of chassis, other types of bacteria that are pre-evolved with some interesting characteristics. And then we take them to the lab and, through genetic technologies, we delete genes, we put genes here and there. And at the end, we produce what you may call an optimal carrier for a given pathway. For instance, if the pathway that we want to implement in the environment needs a lot of reductive power to execute the reaction of interest, then you have to look for bacteria, for carriers that have a background metabolism that will produce also a lot of NADH or NADPH, reactive power to really sustain the reactions. So that's one thing. Also, if the target place is in a hot place, you have to look for bacteria that might be able to support temperature or radiation or harsh conditions, desiccation, all these aspects. So what I find fascinating is that at the end you have to think in terms of a whole process in miniature. It's not only the core process, the chemical reaction, but also have to wrap that in many layers of control, including physical-chemical conditions of the place where you want to release your agent.
And what would you say? Would you say there's particular sort of bioremediation sort of like a specific? What would you use this these sort of processes and these what would you use these processes for? What is it specifically that we want to do in the real world?
Okay, excellent. That's the kind of core issue. So obviously we have to make, it's made already, a list of what are or which are the most important environmental challenges that we face now at the moment. On top of the list goes greenhouse gases, CO2, fluoro-containing gases, methane, and some others. And these are the ones that are more important for climate change and increase of temperature and all these negative consequences of our lifestyle. And so synthetic biology in the last few years, as you may know from the literature, has produced many interesting developments, for instance, in terms of new pathways for CO2 fixation, improvements in photosynthesis, improvements in inhibiting photorespiration, new ways of capturing CO2 in a non-photosynthetic fashion. I mean, there's a lot of interesting pathways and activities that synthetic biologists have developed in the last few years that could be upscaled to some extent. If they were upscaled, it would be fantastic. But there are other questions, not only these gases. There's this big problem of plastic pollution, as I'm not discovering anything new here. Plastics were surprisingly not an issue 20 years ago as a pollutant, and now plastics are being realized as something really bad because they enter the trophic chains and they break havoc in many ecosystems. Well, as you may have seen in the literature in the last few years, different laboratories have identified enzymes with a tremendous potential for degradation of different polymers, and they can be expressed either as isolated enzymes, but also as part of a kind of biological agent able to depolymerize some polymers and use the monomers or the small molecules that are released as substrates for growth. And if they don't do it, you can go to the laboratory and use synthetic biology to develop some type of a trick to make this happen. And so that's another big challenge. And I think that synthetic biology has a lot of possibilities in that respect. But there are other challenges that may not be as evident and to me are super interesting and worrying. For instance, dry lands. So there's this massive evidence of expansion of dry lands at a planetary level. So there are more and more deserts and more, say, dry areas. And well, can we do something about it? And different laboratories are trying to see, for instance, whether we can engineer bacteria that go into the soil and that express on their surface some type of proteins that can capture residual humidity, and in that way to change a bit the kind of position of the tipping points that will make the choice of whether the system will degrade completely or will still recover. So again, for this specific problem, there are opportunities for synthetic biology to really make a difference. And well, then you have micropollutants, then you have a problem with water. I mean, you can make a list of them, but the good news is that if you go one by one, you immediately identify areas where synthetic biology can really do something useful and produce technologies that so far have not been efficacious so far.
And so would you say, but for example, in terms of, like, let's take the example of the expansion of drylands, obviously, because there are a lot of problems, not problems, but like you mentioned, challenges to using these bioremedial solutions in the environment. You kind of mentioned in your article this kind of idea of if there are no alternative solutions. Would you say that, let's say, the expansion of drylands is one of those where there are no alternatives? And how do you think that could impact sort of, let's say, policy around these kind of sort of taking the bioremedial solutions into the environment questions?
Yeah, okay, very good point. So, well, the kind of theoretical background of your question is that not every ecosystem reacts identically to stress. So some ecosystems are subject to what ecologists call linear dynamics. So that means that you put out of stress, the system deteriorates, but if you stop the stress, the system recovers the initial situation. And that's great, because that's the kind of background idea about reducing emissions and everything. And the hope is that if you reduce CO2 emissions and you reduce plastic and you reduce this and that, then eventually the systems will recover by themselves. But there are many other cases that are thoroughly documented in the scientific literature where many ecosystems have gone beyond a tipping point. And once you go beyond a tipping point, just reducing emissions will not help, they will not make a difference because the system has found another stable state beyond the tipping point and it will not recover just by itself. And this is, in my opinion, the window of application of these super kind of sophisticated genetic technologies for remediation, because I don't see any other possibility to revert the state of affairs unless we make these kind of science-based and rational interventions to really go beyond the tipping point in the opposite direction. So, the same problem has been addressed by other disciplines, for instance, geoengineering. So this geoengineering is this idea that you put mirrors to handle the albedo of the sun, or you make super massive installations for capturing CO2. Well, that's another strategy. Problem is that you need a massive investment to do that. And also that these platforms will not self-propagate. You put them in one place and that's it. The advantage of making an alliance with the environmental microbiome is that it accounts for most of the, what you may call, enzymatically active biosphere in terms of biomass, in terms of activity. And therefore, if you think about it and you make some numbers, you realize that environmental microbes and the whole environmental microbiome might be our only ultimate ally to really do something with the climate. Because, you know, in the past, in the history of Earth, microbes have made a difference in terms of climate modifications as well. So, so this is the kind of mental scheme that, in my opinion, could be entertained. Yeah.
Advantage to using then synthetic biology to solve. So instead of and partnering with the biosphere is that we can actually kind of integrate our sort of products with the actual products of the biosphere itself. So could you actually, because you keep mentioning the term environmental microbiome, when you actually consider the environmental microbiome, what does that entail? What's included in the environmental microbiome?
Okay, well, what it entails is what every microorganism in water, every microorganism in soil, and every microorganism in air, you know, and these are like the three major global ecosystems that we have. So they're having calculations of how much biomass is out there in the oceans, in soil, and in the air. And well, the numbers really indicate that if you eliminate just one molecule, that is cellulose or lignocellulose, that is a kind of inert polymer from plants, the bulk of the Earth microbiome is provided by microorganisms. And you know, and this is good news in the sense that we are a live planet and we can, you know, have access to what microbes do because we have access to their DNA and we have all these tools and everything. And well, because of the fact that we have these allies in places such as the ocean, in surface waters, in soil, and in the air, give us a tremendous, say, opportunity to see to what degree we can program them or convince them to execute reactions of interest for remediation.
And you mentioned also sort of in the article, the kind of idea that we have to take our solutions in a sort of like with an Earth systems approach when we want to put them into effect. What did you mean by this? How do you imagine this Earth systems approach to be?
Well, you know, I think that again here the comparison, the illuminating comparison is with medical sciences. You know, so you know, when you have a disease or a medical problem, you have like many levels, many, say, connections, many relations with other aspects, and you have the genetics, you have the environment, you have the heritage, you have the lifestyle, you have many things. So when I talk about this global approach, I think that the environment, you know, the environment is a word that may mean many things at the same time, may mean nothing. But you know, all these aspects that are at stake to influence the environment at some point have to come together, you know. And one of the surprises when I was writing this article, when I was starting to read from places away from my own expertise, is this lack of communication between different, say, stakeholders that have something to do or have something to do about the environment. So, for instance, forest scientists, in many cases, they ignore the fact that in the forest soil you have microbes, you know. So that's completely surprising. And, or, you know, or marine microbiologists ignore this other aspect of molecular biology of cyanobacteria that happens to be there. So you need, or we need, to bring all these expertises together. And that has been done in other areas, in medical sciences, for instance, you know. So one of the arguments that I would like to share with whoever wants to listen is that if we could apply to environmental problems the same effort, the same kind of talent, and the same funding that we have spent in human diseases, you know, many of the problems that we have in the environment will be gone simply because, you know, you have a lot of research that has not been done and needs to be done to really bring up solutions. And this is an important aspect. But what we say environmental problems, I mean, it's a multi kind of dimensional feature that needs to be tackled through many different perspectives. But to do that, we need also to develop the right interfaces and to create some type of a common jargon, common language that allow us to communicate with different types of expertises.
So these solutions really need an interdisciplinary approach. Are you hopeful that we will actually be able to in some way model and have sort of standardized units for measuring all the interactions within the Earth system? Can you imagine that kind of thing happening in the future?
Well, that will happen, we like it or not, because if we don't do it, we are doomed. I mean, and maybe the choice is to be doomed, and then that's it, and no more problem. But I think that if we want to react to the current crisis and the crisis that will come very soon because of the climate change, then we need to develop all these issues that you just mentioned, you know, interfacing, jargon, units, standards, to bring together different disciplines. And also, you know, I think that we need also to kind of leave aside all these precautions and concerns about the use of genetic technology, you know, so, you know, we can spend hours discussing GMOs and these things. And but you know, in these cases, we're not talking about food. We're not talking about making companies to make more money. In these cases, we're talking about solving problems that perhaps there's no other way to solve them but through interventions that involve the release of engineered biological agents. And well, some say communities may say, well, this is crazy. There are many safety issues. A lot of things can go wrong. I agree. Give me other alternatives, and then and then we can start having a good conversation. But the issue in many cases is that you know, there are no other possibilities, or if they are, I don't know them, because I insist that just reducing stress is not enough in many cases. So and unless we do something, things will not happen by themselves. We need to help the planet to really find a solution to the problem.
Do you know of any are there any already sort of or in development softwares or sort of, well, different softwares that do model these different, obviously there's, you know, within a discipline there's software that models specific interactions, like, you know, different softwares that model specific interactions, but are there any that do consider sort of modeling a whole kind of Earth system, or like, let's say, like interactions between sort of like the hydrological, geochemical and biochemical processes in a specific biome?
Yeah, okay, well, right now, you say, is there one kind of complete a tool for making a big model of everything that is going on? The answer will be right now, no. However, there are very interesting initiatives in different parts of the world to do exactly what you are saying. Just keep in mind that, you know, until very recently, having a model of a cell, you know, was a super big endeavor. You know? And just for the last few years, you know, people working with mycoplasma so have been able to have a complete cell with every constant, with every interaction, with every molecule mapped and tracked and everything. So I insist that, you know, if we could apply this talent to the environment, probably the type of models that you mentioned would be feasible. You know, one specific aspect of what you say has been developed in the group of someone called Ricard Soler in Barcelona. He's a theoretical physicist, on what would be the effect of introducing a new member of a community, I mean, a synthetic member of a community. What would be the influence in the structure of the community, how it would propagate, whether it will have positive effects, negative, and so on. So, because, you know, despite years and decades of discussions of the dangers of GMOs, you go to the literature and there's nothing on modeling what would be the effect of releasing the GMOs into the environment for this particular application. So, but this is something that physicists and modelers have to do. Biologists cannot do it. And this is why we have to bring people of different expertises. But, you know, think about it, Sarah. So think all the power to solve environmental problems that we have if we could transfer the effort that has been made in medical sciences to cure diseases, to curing environmental problems. Probably will be in much better shape than we are right now.
So you actually, because I think this is another sort of challenge to incorporating these solutions that you mentioned, which is the sort of the impact of, you know, our sort of solution be colonizing the current, the sort of the environment that it goes into. Could you talk a little bit more about this?
Yeah, okay, well, you know, evidently the starting point is that you have sites with some problem, whether it's pollution or whether it's emissions or you name it, and then you want to introduce an activity. So you have various technical choices to introduce new activity into a pre-existing ecosystem. One of them is trying to enter a new member of the in the biological community, a member that you have developed in your laboratory. Try to make it a new member of the place where other members are. This, I have to say, has not been terribly successful, because in ecology, there's this phenomenon that has been known for a long time that is called resistance to colonization, or colonization resistance. And that means that once you have a well-formed community of different partners with interactions and chemical transactions and everything, they really develop a mechanism or various mechanisms that prevent the entrance of a newcomer. Okay, so there's massive literature in the late 80s and early 90s of attempts, for instance, to introduce a recombinant bacterium with some interesting genes for bioremediation in a pre-existing community, and in many cases it did not work very well. So the alternative that we are entertaining, and this is in fact one of the major projects in my laboratory, is to see whether, instead of introducing a new member, what we do is that we enrich, we fortify genetically the community with a new activity through, for instance, engineering horizontal gene transfer. Okay, so what we do, and we have a large project in my laboratory on that, is that we go to the lab, we engineer the pathway of our choice, and then we put that in a hyper-promiscuous plasmid, for instance. And then the plasmid is engineered, and I can give you details on how to really go everywhere and to get expressed everywhere. And in that way, we think that instead of having a new member of the community, what we do is that we allow many members of the existing community to develop a new activity. And that's something that ultimately, in my opinion, could do a better job than just trying to, you know, to colonize the place with a new member that sometimes the residents are not very happy about.
So in some ways, this is like a step even further into the direction of let's keep working with the system we have rather than push against it, right?
Absolutely, you know, and you know, I have developed in the last few years this idea after working for many years with this type of problems, that it's really difficult in the environment to force bacteria to do what we want them to do, you know. So so at the end, there's evolution, there's mutations, and the system will collapse very quickly. So I think that we have to kind of understand a little bit better the lifestyle of environmental bacteria, and even I would even argue that we need to start a conversation with them in terms of partnership, not in terms of domination, you know. So every time we try to dominate the bacteria, it doesn't work, you know. So because the microbial world will lash back in terms of mutations and evolution. However, if we understand, you know, and we kind of establish some kind of partnership with them and understand their lifestyle and their needs and everything, maybe we can convince them to really express these new activities. And so, and I think that this way of enriching and fortifying existing microbiomes with new useful things might be ultimately better than just, you know, forcing the community to adopt a new member. Okay. And
in terms of, so what bacteria do you work with in your lab?
Well, you know, as I said, you have to make a choice of chassis, and depending on the ecosystem that you are interested, you have to pick one or two or whatever. So our favorite bag for these applications has been the soil bacterium that is called Pseudomonas putida, and this is a bacterium that have been working for a long time. Is agram-negative. We have developed many tools. Well, we and others have developed many tools. And now is one of the pets, say, for showcasing the power and the applications of synthetic biology for this type of issues. I have to say also that because of the properties of these microorganisms, then Pseudomonas putida is good not only for bioremediation and for these release applications, but also as a chassis for industrial biotransformations. And one of the reasons, and I mentioned that on passing before, is because the native metabolism of Pseudomonas tritida is pre-evolved or pre-geared to produce lots of reductive power. And that simplifies a lot both degradation reactions and also synthesis reactions. So this is a super nice platform. We're very happy having working with this bag for a long time.
So in some ways we need to have a lot of knowledge about diverse species in order to be able to actually create the solutions that we want. So in terms of, I guess this question comes very late into the conversation, but if you were to sort of explain, like within a you mentioned this kind of term, the bioremediation unit, I think. What do you have in a bioremediation unit?
Okay, well, you have to go case by case. Okay, so but one possibility of a bioremediation unit would be to engineer a formulation involving not just one agent, but involving various agents. And I think that, for instance, one very interesting combination would be to put together a plant and a bacterium. Plants can capture CO2 from soil and then can release some carbon into the soil, and the carbon may become the substrate for maintenance of a bacterium that has been engineered to degrade something. Okay? So, one possible unit, for instance, would be to have a seed of a plant able to grow in a specific location. Maybe the plant has itself been engineered to release given carbon sources or to have some specific root architecture. And this is something that plant molecular biologists can do. And then to coat the seeds with a bacterium that itself is engineered to degrade this or that, or to fix CO2, or again, you name it. And then once you plant this unit, then the prediction, you have a kind of nice, a partnership between the plant and the bacterium that will sustain a catalytic activity in the soil, for instance. That would be one possibility. But you can think on others. For instance, you can think on engineer a small microbial consortium, everything just microbial, for instance. You put it in a gelatin capsule, and you make a formulation in such a way that you put different partners in a complex reaction. One partner takes care of getting rid of a stressor. The other will capture a bit of humidity. The other will release some protective agent. And also, in cases where you don't want this agent to spread too much, you can even think on putting in the formulation some, compound that is necessary for the growth of the bacteria that are inside the formulation, in such a way that once this is consumed, the bacteria stop growing. You can think on this type of ideas that we can leverage a lot from the expertise in pharmacy. Because I insist that at the end, the problem of giving the environment a remedy for a problem is not completely alien to the problem of giving a drug to a sick body. So maybe there are many concepts and many conceptual frames that you can import from this kind of pharmacy into environmental scenarios.
And of course, we spoke about sort of this need to sort of solve like environmental stresses by when there is no alternative using these solutions. What do you think about this idea of sort of maybe reimagining how our environments look like? Can you ever imagine these kind of procedures or these kind of ideas in forming actually a reimagining how an Earth would look like, for example?
Absolutely. That it even goes into the kind of essence of synthetic biology, not only to provide technical solutions to think, but also to imagine how this solution can bring us to a kind of different scenario and to a different covenant with the natural world. And that in the community of synthetic biologists, there are not only molecular biologists or engineers, you have also a bunch of people with artistic, background or philosophical, humanistic, whatever, that try to see to what degree these technologies can take us to a new world and to a new type of civilization and a new type of partnership with nature. And well, you know, in the laboratory, we have had recently a wonderful experience with an artist in residence. Okay, and so this is something that is becoming very popular, at least in European projects. To have someone that comes from a different perspective and then spends some time in the laboratory, talks to people, and then the person proposes something that is connected to the work, but also connected to kind of anticipatory vision of what would be the consequences. So we had a wonderful experience, and we came up with a couple of projects in which well, she was a very interesting plastic artist called Lara Tabet, and she proposed, you know, things that to us were, wow, this is super interesting. Ways of interacting directly with bacteria by making interfaces between our talk and the signals that bacteria use to communicate to each other. Well, I may not give you all the details now, but you know, yes, this is an idea that we can communicate to some extent with the bacteria, and also have this other project on how to use this propagation of genes in a wider microbiome to really even change the mood of an entire population, in terms of being more optimistic or being, you know, because as you know, the microbiome has a big activity in our mood and everything. So, I mean, this is an artistic performance, okay? So we did not do it in practical terms, but at least as an exercise of anticipation, it's interesting and provocative to think to what extent these technologies of thinking big about the microbiome and so on can really change our perspective of a different future, right?
That's really, really incredible. I didn't know about this artist. I knew that some labs do have an artist in residence, but I didn't know that it's becoming more popular. That's really quite fascinating. I really like how it takes, you know, when people want to lose weight, it says, oh, don't change your behavior, change your environment. And then it's here, change your environment, change your mood. That's really incredible. I really like this. Yes. I wanted to also ask you about, so when you, well, it's kind of a semantics question in some ways. You mentioned terraforming motifs in your work, in your article. Can you explain more about this?
Well, the concept of terraforming motifs come from this laboratory that we collaborate with in Barcelona. And this is this notion of how we can, in the laboratory, say abstract and model what will be the minimal activities and the minimal interactions that you need between different biological agents to start having a kind of agent, a kind of instrument to create a new environment if delivered to a place where before there was no life. So, well, this is a theoretical question that comes from the times of, well, still today, NASA and astrobiology and how to bring life to Mars and all these places. And, you know, the basic question is, how do you bring life to a place where there's no life? And the argument is that, well, we don't need to go to Mars. I mean, we have many places on Earth that are in super bad shape. How can we bring a new life to these places? And the theoretical problem in many cases is the same. And the starting point is to first make a list of what will be the minimal set of interactions between different biological partners that will be necessary to have a self-sustaining niche creating system in either Mars or in Earth, and that's the kind of a scheme where you have the components and interactions and the logic gates and the logic and everything is what in theoretical, say, models is called a motif, a terraforming motif. But I insist that this is more the work of our collaborators in Barcelona. But this is super interesting because at the end, it's not enough that you put just one bacterium or one agent. You need to have a kind of self-sustainable consortium that can really prosper by itself. And to that, you need to engineer interactions and other features as well.
Trying to answer the question, so like you mentioned, sort of, how do you bring life to somewhere where it's lifeless? That's very, very cool. So what are your, sort of, in terms of your lab, you mentioned the sort of hyper promiscuous bacteria that you're engineering. What are your next projects, your sort of like the next steps sort of for the future?
Okay, so, well, I'm very interested in bringing to the laboratory the possibility of creating new activities, you know, and you know, when you make the list of environmental problems, you say, well, we have this desertification, we have this problem, this other and this other. And in some cases, or in most cases, you may figure out some biological solutions. But in other cases, you say, well, we don't have a good activity that has evolved already to do a good job here. For instance, plastics. Plastic is a super important issue. So when you look at the sequences of the enzymes that are involved in plastic degradation, what you observe is that you can make a lineage, and then you can identify that these enzymes come evolutionarily from other enzymes that have mutated here and there and have acquired new specificity or new activity. So that means that in this case, we have an evolutionary case of innovation. So, but I wonder, and this is something that I would like to bring to some experimental platform, whether we can create, an evolutionary platform that creates authentic novelty. And novelty is not to take something that exists and modify it a little bit to do something else, but to start to create new activities from scratch. And this is something that we're very excited and well, we think that we'll have some ideas on how to bring it about. So I think that to have a list of activities that are interesting for environmental applications, you need to rely on things that are already there. You have to rely on things that can be created through innovation of existing things, but also there's this big opportunity to kind of domesticate evolution in the laboratory, and that has been done to some extent in the past with different approaches, and see whether we can have biological systems that can create novelty, authentic novelty in terms of enzymatic activities and other activities of environmental interest.
So when you say, so in terms of creating novelty from scratch in this platform, do you mean like a machine learning platform or a very biological platform?
Well, that will require some other podcast probably, but, basically the idea that we have is to create a biological system able to process information and to accelerate a lot identification of solutions in a dense solution space. So it's a combination of computation and weight experimentation. And we know a lot now on how bacteria evolve and what are the major mechanisms for creation of diversity. And our kind of idea is to explore whether we can bring many of these together and really amplify tremendously the evolutionary capacity of a system that we can have in the laboratory. So that would be a kind of sketchy description of what we would like to do in the future.
I will be looking forward to the research coming up from that. That sounds really incredible and really very worthwhile to do. So I think my final question to you is, obviously, you started writing this article and you started reading outside of your own discipline. What actually inspired you to write this article? What inspired you into this field?
Well, I'll tell you what inspired me. So years ago, I was part of a working group in the in the World Economic Forum, WEF, in Dubai on biotechnology. And we have discussions. The group involved biologists and biotechnologists and so on and so on. But then at some point, we had a meeting with a similar group of the chemical industry. And then it was like a revelation, So we started to talk and they said, oh, we can program bacteria to do this interesting compound and to make biofuels and to, make these sophisticated molecules and to create materials for textiles and to create materials for construction. And this is wonderful. And the chemist said, well, that's fantastic. How much can you produce? I said, well, we can produce 3 milligrams of this compound in a liter of culture. And they went to laugh, So they started laughing. I said, well, look, you are very nice people, very enthusiastic. When you discuss tons and millions of tons of something, you come to me and we keep on discussing. Okay? So that came to me. I say, well, the big issue in synthetic biology is scaling up. I mean, there's no question about that. You can make the most wonderful things in the laboratory at a small scale. You can have papers in the most wonderful journals, but if you cannot scale it up, your efforts are completely useless. And scale up, is something that engineers have dealt with a long time ago, but biology requires a kind of thinking and a kind of approach to scaling up that is very different of scaling up, other industries. And you know, an environmental problems is one of the super important case where unless you scale up at a very big dimension, the solutions that we create in the laboratory, I mean, what we do is completely useless. So I think that we really need to convince the community that it's wonderful to do things at a smaller scale, but we need to fill this gap between the smaller scale and the bigger scale, even thinking big at a planetary scale.
Right. Thank you so much for joining me today.